In this video, we will be talking about what the urea cycle is, where does it take place, why, and how. Urea cycle is a cycle of biochemical reactions that produces urea from ammonia. Urea cycle takes place predominantly in the hepatocytes of our liver cells and also in our kidneys. It is important to transform ammonium, which is a toxic byproduct of amino acids, into urea, which is a less toxic form.
Now, let's talk about the cycle. We can break down the cycle into 5 steps. The first 2 steps take place in the matrix of mitochondria of the liver cells. Meanwhile, the other 3 steps take place in the cytoplasm of hepatocyte of liver cells. Before we explain the urea cycle in depth, let me show you how we can remember the intermediate steps and the enzymes involved easily. For the intermediate steps, remember the sentence, "Our caring citizens are always furious about public urinating." O. Ornithine, C. Carbamyl phosphate, C. Citrulline, A. Aspartate, A. Argininosuccinate, F. Fumarate, A. Arginine, and U. Urea.
For the enzymes involved, imagine the sound of a crying baby that got scared by seeing people urinating publicly. Where is the sound that they make? It can be represented by the letters COAAA. C for carbamyl phosphate synthetase, O for ornithine transcarbomaxylase, A for argininosuccinate synthetase, A for argininosuccinate lysase, and A for arginase.
Urea cycle begins in the matrix of mitochondria of liver cell where the combination of carbon dioxide and ammonia produces carbamyl phosphate. It requires 2 ATP and also requires enzyme carbamyl phosphate synthetase 1. This enzyme requires N-acetyl glutamate in order for it to be activated. After the formation of carbamyl phosphate, this combines with ornithine that leads to the formation of citrulline. This reaction is catalyzed by the enzyme ornithine transcarbamylase. This step will occurring in the liver, specifically in hepatic mitochondria. Everything else except for the first and 2 steps are going to be occurring in the liver but not in mitochondria, rather it will be occurring in cytoplasm. Now in the next step, the citrulline is transported into the cytoplasm. It combines with aspartate and ATP leads to the formation of argininosuccinate. This reaction requires 1 ATP molecule and is catalyzed by the enzyme argininosuccinate synthetase.
In the fourth step, the enzyme argininosuccinase acts upon argininosuccinate, producing 2 molecules, fumarate and arginine. Fumarate acts as a bridge between urea cycle and gluconeogenesis that can be used by liver to form glucose. The last step in urea cycle, arginine will go back to ornithine, but in the process will convert to urea. Enzyme arginase acts on the arginine to produce urea and ornithine. The ornithine will be utilized back in urea cycle, whereas the urea produced will be dissolved into the blood and is transported to the kidneys to be excreted in the form of urine. So in the urea structure, we can see that the carbon comes from carbon dioxide in the first step. Remember that carbon dioxide in our body is actually in the form of bicarbonate. Next, the oxygen is from water in the 5th step. Then, the first amino group is from ammonia in the 1st step, and lastly, the second amino group is from the aspartate in the 3rd step.